start with an animation on flexion and flexion pathways. Let's see. That's the one I want. Here we go.
The spinal cord is also the final common pathway for most voluntary and involuntary reflexive movements. In this animation, we'll examine the spinal pathways involved in the withdrawal of a limb from a painful stimulus. The cell bodies of sensory neurons are located in the dorsal root ganglia. Sensory axons enter the spinal cord through the dorsal roots, where they synapse upon interneurons, which in turn project to the motor neurons. The sensory receptors that transmit a sensation of pain are called nociceptors. Motor neurons are located in the ventral horn of the spinal cord and send their axons out the ventral root to innervate muscles. Some motor neurons innervate flexor muscles, while others innervate the extensor muscles. Whether a particular motor neuron is excited or inhibited depends on the interneuron. In this example, an interneuron sends an inhibitory signal to motor neurons controlling the extensor muscles. An excitatory signal is sent to motor neurons controlling the flexor muscles. This permits the dog to withdraw the hind limb from the painful stimulus. With the withdrawal of one limb, the weight shifts to the contralateral limb. Interneurons also connect to motor neurons on the opposite side, so that the muscles of the other limb can adjust to the shift in weight distribution. Flexion of the stimulated limb is therefore accompanied by an opposite reaction in the contralateral limb. That is, the extensor muscles are excited, while flexor muscles are inhibited. The sensory information is also relayed up the spinal cord to the brain, where it can be further processed. Ultimately, signals from the brain travel back along motor pathways down the spinal cord, and can thus modify the actions of the spinal circuits. The information from higher nervous centers permits fine-tuning of muscular control, and also allows the dog to avoid painful stimuli in future encounters.
He's a happy dog. Okay. So think for just a minute about the difference between two different walking scenarios. One, you're walking across campus. It's between classes, there's not very many people around, and you're just walking straight across the quad on the cement. Okay? Pretty simple. Two, you're hiking in the mountains on a rocky trail with lots of stumbling blocks and trees and bushes that are in your way and things like that. What's the difference in those two scenarios in terms of central involvement in walking?
Probably in the second scenario, you said central, you actually have to cognitively think, rock, tree, how am I going to get out of the way? Well, I guess I kind of step over here, kind of over there.
Yeah.
As opposed to walking on campus, there's nothing really, no obstacles.
Yeah, in fact, the more difficult the trail, the more close one gets to actual cognitive involvement in planning walking. I mean, think of the last time you hiked a really difficult trail where if you didn't think about where each foot was going, you might fall. Well, you were probably consciously thinking, now what would be best here, this rock or that rock, or is that a slippery spot? You were probably actually thinking about what you were doing with your feet, and these pathways from the brain into the brain were probably much, much more active in a situation like that. Even one where maybe it's a little less difficult and you do have to negotiate the trail, but can do it while you're talking to your hiking buddy and not really think about it, right? Whereas when you're walking across campus, very little brain involvement is required. It's pretty much central pattern generators in the spinal cord that are controlling left and right and left and right. And you might think, I'm going to be late, I better speed up, and that might change the cadence of those movements. But basically, there's not a lot of brain involvement in what's going on in that kind of movement. Or so.
as the stretch, deep tendon, or myotatic reflex, and it is the basis of the knee, ankle, jaw, biceps, or triceps response tested in a routine physical examination. These reflexive responses are too rapid for involvement of the brain and are mediated by the local circuitry within the spinal cord. The sensory signal for the stretch reflex originates in muscle spindles, the sensory receptors embedded within most muscles. The spindles comprise 8 to 10 intrafusal fibers arranged in parallel with the extrafusal fibers. The extrafusal muscle fibers make up the bulk of the muscle and are innervated by alpha motor neurons. There are two structural and functional classes of intrafusal fibers: the nuclear bag fibers and the nuclear chain fibers. These fibers differ in the position of their nuclei, the intrinsic architecture of their myofibrils, and their dynamic sensitivity to stretch. Large diameter sensory axons, group 1A and group 2 afferents, are coiled around the central part of the intrafusal fibers. These afferents are the largest axons in peripheral nerves. Because action potential conduction velocity is a direct function of axon diameter, these fibers mediate very rapid reflex adjustments when the muscle is stretched. The stretch imposed on the muscle deforms the intrafusal muscle fibers. In the afferent axon endings coiled around the spindle, mechanically gated ion channels become activated, initiating action potentials in these neurons. Group 1A afferents, which preferentially innervate nuclear bag fibers, respond phasically to small stretches, while Group 2 afferents, which innervate both fiber types, signal the level of sustained stretch by firing tonically in proportion to the degree of stretch. Small gamma motor neurons modulate the level of excitability of the muscle spindles. The sensory neurons form monosynaptic excitatory connections with the alpha motor neurons and the ventral horn of the spinal cord that innervate the same, homonymous, and synergist muscles. The sensory neurons also innervate local circuit neurons that form inhibitory connections with the alpha motor neurons of antagonistic muscles. The monosynaptic pathway from the spindle to the alpha motor neuron is unusual. Most sensory neurons from the periphery do not contact the lower motor neuron directly, but instead exert their effects through local circuit neurons. When the muscle is stretched, the afferent volume...
So whenever you see a very simple connection, you can probably infer that speed of conduction is important in that connection. Let's kind of go on.
Volley from the muscle spindle is relayed to the alpha motor neurons, and the efferent volley returns to the muscle. Since muscles are always under some degree of stretch, this reflex circuit is normally responsible for the steady level of tension in muscles called muscle tone. When the muscle shortens, the spindle is relieved of tension, or unloaded, and the sensory axons that innervate the spindle might therefore be expected to stop reporting information about muscle length. However, gamma motor neurons become activated at the same time. These neurons terminate on the contractile poles of the intrafusal fibers, causing them to contract, thereby maintaining the tension on the central region of the intrafusal fibers where the sensory axons terminate. Thus, co-activation of the alpha and gamma motor neurons allows spindles to function at all muscle lengths during movements and postural adjustments.
So what's going to be the overall, what's going to happen because of this intricate circuit? How does it contribute to the natural state of our muscles? Any thoughts on that? And why do we need it?
Because otherwise our muscles become functional constantly and we wouldn't be able to relax. If they're constantly contracted, then they're really used to them.
Yeah, so
You can't contract them anymore.
So no matter what position your body is in, you have a sort of constant level of muscle tone. Now, if you're someone that lifts weights, that constant level of muscle tone might seem increased. The muscles might have more tonus to them. However, all of us have muscle tone constantly. So when we move, or when gravity, maybe causes us to move, there's no damage that occurs to muscle because the muscle length automatically adjusts and maintains a constant level of tone, no matter what we do to it, whether it's stretched out or whether it's sort of bunched up. So it prevents damage to the tissue and maintains constant tone so that from whatever position we are currently in, we can quickly contract. And our initial message from the brain to contract is not going to correct any sort of over relaxation in the muscle. It's always ready to go. There's always some tone in it. Okay. Well, we'll take it up from here when I see you on Tuesday. Yeah. You do maintain muscle tone while you're asleep, yeah. Otherwise, your muscles could get damaged. They have to be sort of able to adjust to whatever position you're in. Yeah, good question.